Yes. So please let me know once you are able to see the slides. Anyone, if you can confirm me. I have to maybe check. Check. No. How do I know that screen is visible or not? If someone can unmute and can tell me that screen is visible. Please speak up. Okay, sir. Okay, thank you, dear. Thank you. I will also show you the one or two practical examples that so that you can by yourself can download a protocol, right? Because that's more important. So stay till end. Let's together we will learn what protocol is. So now if you look at clinical trial industry or clinical trial, so clinical trial journey is like there are a couple of steps. Like first is our planning. That is number one step. So because this is a complex thing, we are testing our drugs on a human, so planning is important. You may agree with me, huh? Then based on this planning, whatever the planning they have done, they conduct a trial that you see at a hospital. Hospital mostly they or in our wording we say site. The trial is conducted, and when they're doing this trial, then data is collected. So now whatever the data they are collecting, they write a report. Basically we are coming to end, right? So these are the three basic part of a clinical trial. So now different functions in a clinical trial, they get their job roles come into picture. Like a planning, medical writing plays a major role because you have to write all the documents. When your trial is ongoing, you are doing the trial, your role is maybe little less. Maybe there is an update, maybe there is an amendment done. But operation team, like you are a CRA, CRC, even data management, right? Their role is high, even PV associates. Once trial is over and the data is here, SAS team has analyzed the data, right? Statisticians, they have analyzed the data. Closer to the end of the trial, you also have to write a CSR, narratives, maybe write the publications, right? So end of the trial, also your role is high. So this is on high level, but our topic is protocol development. Now, at a planning, there are a couple of documents those are required, right? One of that is investigator brochure. Second is protocol. Protocol, even we write together ICF, informed consent form. When you will be working in a medical writing team, you will realize that informed consent is almost prepared along with protocol, because most of the content in informed consent form is from protocol. Then another document, maybe you will be needing your SOPs, maybe manuals, right? Lot many different work instructions, lot many different documents are required. But this is primary document. So at a planning stage, protocol is important. At a closest stage, CSR is important document. So what is protocol means? Plan of study. So whatever is written in the protocol, that you will be doing. So protocol is plan of the study, or you can say the blueprint of the study. So try to understand in a simpler language. I am very confident that you must have done lab experiment in your graduation, or maybe if you are doing master, you're still doing some experiment. So protocol is like your instruction. all the instructions are given in the protocol. What all you need to do in a clinical trial, that is written in your protocol. So any question on what is protocol? So here is a definition. So what is the goal of the protocol? I have already told you that it is an instruction document. It is a plan of the study. So this is the document that describes the goal, design, methodology, statistical consideration, and organization of a trial. Objective is goal. What you want to do? Design. How you will do? Statistical consideration. At what stat method you will be applying. Now I will tell you that all this you need not to do by yourself. You will be getting help. So it should be scientifically sound. And it is submitted to the local national registry for approval. So I have also, maybe in initial lectures, you must have learned IND or NDA application, right? One of important document in this is protocol. So regulatory body, they will see your plan. Based on this plan, they will either say yes or they say no. So this is submitted to your local, national, or international regulatory bodies as required, right? What else for you as a study team who is doing trial? It is an instruction document. Now, what other benefit is for the trial is on 10 sites. It is on 10 places. All will be following the same plan. We are having only one plan. Plan are not different. So what is the benefit? There will be no deviation. So everyone will be doing the same thing, right? So this is the purpose of the protocol. If someone asks you in an interview, it provide justification that why you are doing a trial. Why? Scientific rationale. We will learn also. Scientific rationale. When you will be doing, when you will be starting, how you will be doing, design. Like number of patient, example. What drug you are giving. I will show you also one real protocol so that you can relate. I will also help you that how you can download these documents, right? So pre-specified objectives and hypothesis. This is instruction to study team exactly what to do. If it is written on day 1, dose 1 will be given. It will be followed like that. On day 20, dose 2 will be given. On day 25, blood sample will be collected. So it will follow the same order. Suppose you are not able to give a dose to this patient was not there. So it will be deviation. That it is written in a protocol, but we are unable to do. So that is a deviation. So exactly what to do, it will be mentioned here. Describe how to treat and manage the subject. Suppose some subject fell sick, SAE happened. This protocol will be having a plan how to handle that. Describe data to be collected and collection method. How you will be collecting data in the form of case report forms. What are the time points like day 1, dose 1? That means you will be giving dose 1 and collecting a data. Describe the plan for data analysis, which SAS programming you will be using, what stat method you will be using, right? So all will be detailed in your protocol. Reference tool. Even what type of monitoring and auditing you will be doing. When suppose once in a week. So once a week. If trial is very complex, once a month. Auditing 2 per year, 2 audits per year. So all plan that will be mentioned in your protocol. So protocol tells you exactly what you have to do. So now, because I have given you, I'm also trying to give you the insight into clinical trial industry. So see, I have given you here study design, methodology. So you may get a question that how you as a pharmacist or as a microbiologist or a biotechnologist, how you are going to draft this whole protocol. But the whole point of my this slide is to tell you, you are not alone. You will be getting a support. You are just a writer in this. So that, my point to tell you is that don't get scared that medical writing is difficult. So here it is. Medical writer will be a central point. Is there any question? Okay. So medical writer will be at a center. So this is just for a reference. You will be having a team of a doctors or a researcher. Suppose the study is on a PK. So you will be having a PK scientist who will be telling you that do we need to assess a Cmax or Tmax or half-life, whatever. What PK parameter we need to include. The PK scientist will be telling you. At what time frames the patient need to be checked, so the doctor or a physician will be telling you. Suppose there is a question related to patent, that is patent protection, right? Legal, basically legal. Patent attorney will be helping you. Investigator, the one person who is going to do the trial in actually on a site and hospital. They will be telling you that is it possible from a practical site or not, right? Study stat, what method you will be using, in your protocol, how you will be calculating the sample size, what is the hypothesis, right? All that will be given to you. Guys, sorry, please put yourself on a mute, because it disturbs everyone, it breaks the rhythm as well, right? So what I was talking about was sample size, how many patients you need, 400, 500. It is always based on some assumptions. Statisticians, they take our assumptions like, okay, let's assume 100 patients for a phase 1, maybe drop rate will be 10%, so 90 patients will be left. Are these 90 patients sufficient, to collect required data? So for these stats help you. So my point is that you as a medical writer, you just have to raise a query. Rest, you are having a team to assist you, to help you, because they are responsible to develop a protocol. Now, you must have seen here I have written sponsor. I have written a sponsor POC. So what happened? If you are working in a pharma company like a Pfizer, Novartis, Novo Nordics, then this sponsor POC will not be there. But why I have kept it intentionally? Because many of you will be working, you will get an opportunity to work in a CR, contract research organization. So there will be always one person from a side of a pharma company, right? So if you need some help, some team is not responding. Suppose this PK scientist is for pharma side, but they are not responding to you. That happens a lot. So you will escalate to this POC. Or other way, POC will also be tracking that whatever the timelines we have, drafted or developed, are you able to deliver on those timelines? So they are the bridge between CRO and a pharma company. So this is now in case you get uh, an opportunity to work as a freelance. Although, when you are a fresher, I always recommend try to get a job initially, then only work as a freelancer. But my idea is that you never know what circumstances happen in one's life and they get into a freelancing. So then also you have to work with all these people. You are not going to work or develop a document alone. Okay, so I'm exactly giving you the template. So just imagine you are working in a CRO or a freelancer or maybe in a pharma company, anywhere. So how you will start? I'm taking a little pause. How you will start? And this is also going to be helpful you in your in your interview. So just imagine, suppose we have to come for this class. What is needed? You as a student, you needed paper pencil, maybe your iPad or laptop, mobile, whatever, right? That is your preparation. You wanted to sit in a silent room. That is your preparation. For me, I need a slides. I need to prepare a material. I need to also sit in a silent room, and explain to you. I should be having that energy. Similarly, for a protocol writing, first of all, as I always explained you, we never ever work as a medical writer with a blank paper. No, that is the beauty. That is a blessing. You are always be having a template. Template means where introduction will go, where background will be written, and not only that, what will exactly go into that. Synopsis. Hmm? I will explain to you as that as well. Investigator brochure. For a protocol, if someone asks you what is source document, IV is investigator brochure is a your source document. Sample size information, small idea, any data available from a previous study, any previous protocol. Suppose you are writing for a phase 2, ask for phase 1 protocol. Suppose you are writing for phase 1, do they have done some micro study? Ask for. Otherwise, animal study data you can ask. Any previous publication, any disease guidelines. Suppose you are doing a trial on HIV. Any guideline related to that? CSR synopsis. Suppose you are working on phase 2 and for phase 1 CSR, you can ask for it. Then you arrange a meeting. And this is the standard for all the medical writing documents. Even if you are getting a work as a freelancer, say that, can we have a 15-minute call to define the scope, what you want to do, and set the expectation and the timelines. And important for you as a medical writer, always document these things. How you will be documenting these things? By meeting of minutes. So ensure, see, if you are working in a CRO, then you have to give it to a sponsor. But even if you are working in a pharma company like I am working, so we are always having in-house kickoff meeting. We always send them some Excel, some other piece of paper, document where the timelines are defined. It may be tentative timelines, but timeline defining is very, very important as a medical writer. Medical writer, job I will say is easier. It's not very difficult the job. But the important here is you are always having a deadlines. So you have to manage that. You have to show some discipline. Also, you have to link up, get coordinate with study start for a sample size calculation or estimate or what analysis method they are going to use. Request clinical team, what are the goal of study, what are the end points and eligibility criteria. So these are the basic information you take from study team. Even, there is a trend in a medical writing world, proof of concept. This team, clinical study team, they generally first draft proof of concept. Basically, goal, endpoint, eligibility criteria, they mention it briefly, just to take the feel of the trial. So you can ask them this document that, can you please give me a proof of concept? So now let's start how authoring of a protocol looks like. So this is a very strong slide, a good slide, I will say, and that you can make a notes from this slide. First of all, you have to work on a strong introduction and a background and a rationale. Now, three things are important in this. What we have learned in GCP principles, ethics, right, and right safety well-being of the subject, that is addressed in here. Even scientifically sound study, that all three principles will get addressed here. So first is, what is disease and what is unmet medical need? You have to do the literature search. Bit of help you will get from a study team, because they are going to give you the disease, and in small, a very brief narrative that what is unmet medical need, why you want to do this trial. Suppose you wanted to do this some drug related to insulin, like recent example is MP, where it is helping, how it is different from other drugs, right? Brief description of what currently know about the drug and disease condition, background, published and unpublished information. Like, these many people are suffering from this disease. Current available options are these, and our drug is, we are trying to do it different. Why you are doing this? Justification. Because just imagine, clinical trial you are doing on human. If your trial doesn't have any meaning, it is not helping society, then there is no need of this trial. The focus on research, question, justification, the trial, overview of study drug, previous result, and its safety and efficacy. This is also where you will also give a hint of your studies that you have done on animals. How you are going to do? Are you going to compare it with some drug which is already available? What dose you are using? What is the justification of this dose? Or maybe you are going to, what you say, refer some animal study that we have used this dose, and it has been found there safe, because that was your biological model. Route and administration. What schedule? Once in a day, twice a week? Is it IV injection, or maybe a once in a week? Whatever it is that you have to give. So what, why, and how you have to answer in your introduction and background. Now, another important thing in a protocol is study population and eligibility criteria. Eligibility criteria is admission criteria. Like, when you go for your pharmacy program, there is an admission criteria. You must have been 60% in your 10+2. You must be studied like a biology or something like that, right? Similarly, who can participate in this clinical trial? There are different examples. Suppose it is a phase 1 vaccine trial. Your population will be healthy volunteer. Suppose it is a phase 2 hypertension trial. You will be taking actual patient, right? So, lot of information on this. Hint is always given by the study team. Because if the eligibility criteria is very stringent, very strict, then you won't be able to enroll your subject, and it won't be a valid clinical practice. It make your study very complex. That means you are setting for a failure. Your protocol is not a good protocol. So what resources you can refer? Previous protocols, clinical trial registries like ct.gov, excellent source. CTRI, EU CTES, European Union, right? Current study synopsis. These are resources you can, I will walk you through this. One I can do, CTRI you should doing by yourself. EU CTES you should be doing by yourself. Recommendation, what is the recommendation when you are drafting eligibility criteria? Keep the number of eligibility criteria minimum. It should not be like you have listed 100 of things. No, try to keep 10-15 inclusion, 10-15 exclusion. Like you wanted to recruit healthy volunteer between 18 to 60, both male and female. So anyone less than 18, it will be excluded. They can't participate. Anyone above 60, they can't participate. A person came in, he's saying, I am having a frequent headaches, can't participate because that person is not healthy. Include only those absolutely necessary for a scientific validation. That's why we have a sample size. It's not like you can recruit 10,000, 50,000 as you want. Like I will having more data. I will get more data means more information, right? No, that's not possible in a clinical trial. Now, study design and randomization and blinding. Study design, you have to define, is it a prospective study? Clinical trials are a prospective. Suppose phase 1 to phase 4, right? But if you are doing some observational study, because you also may have to write a protocol for observational study, then it may be some based on some literature search or a retrospective study. Okay. What type of randomization, non-randomization you are using? What is blinding method? Open blind, single blind, or multiple blind? Arms. Arms means treatment. Am I giving only one treatment, which is say as a treatment, or I am comparing it with B, or I am comparing it with the placebo? So it may have, so if I am giving treatment A, B, and placebo, that means 3 arms. Right? So how you are doing randomization? 99.9% it is by the computerized program, IPRS. How they are generating sequencing, what type of randomization they are using, how they are going to ensure that uniform randomization. Again, you will get a help from a stat. Blinding, what is your criteria for a blinding? Single blind, double blind. What dynamic and predictable and can be changed? How someone can go for unblinding? Suppose some emergency happens, how they can go and interim analysis. Very important criteria. So what they are saying that you need not to wait for end of study. Suppose you are aiming that's how we have done our COVID studies. Suppose you are aiming for 400 subjects. The moment you have a data of 100 patient, you do the analysis and then make a decision that is it safe for a patient or not. If not, you can close your study early. So that is interim analysis. So midway you are again analyzing. Study goals and endpoints. Goals should be SMART. SMART means they are specific, measurable, acceptable by audience, relevant to clinical studies, and technical. Suppose vaccine trial, phase 1. What will be the goal? I'm drafting with you. What will be the goal? Safety. Safety for, just imagine, safety for a patient. That is the your goal. Now, how you will measure endpoint is known as how you will measure number of adverse event and serious adverse. This is how you will be measuring it. Another goal will be immunogenicity. Example, immunogenicity. From then, how you will be measuring immunogenicity? Change from baseline to end of study. So baseline means day 1, or you, in our language you say day 0. Day 0 you have given a, before a vaccine you will see the antibodies, right? And at the end, after 2 doses, again you will see the antibodies, and you will compare with this. How much it increased? If it is increased, that means immunity has increased. If it is not, then immunity has not increased. So that is how you set the objectives and the endpoints. Any question till now? Because it may sound a little technical. Okay, I hope you are able to understand, right? Even if you are not working as a medical writer, this protocol is important for you. Because suppose you are example in the operation team, suppose you are working as a data manager, suppose you are working as a pharmacovigilance associate, protocol is unavoidable document. You should know it. So now the methodology. So we know who can participate, we know what to measure. Now how you will measure it? How you will measure? We said immunogenicity. We said serious adverse events. What will be the method? How you will be collecting this data, right? So how many sites you will be including? Like, is it international trial or a local trial, multi-centric trial? So that will be answered here. Screening and recruitment and enrollment procedure. How you will be recruiting a patient? Recruitment strategy. Say example, one recruitment strategy I can share with you. Suppose you wanted to work in, you are working in a public health. Maybe you are working in the ICMR. You are working in a TB study. So what will be your recruitment strategy? Maybe you will be sending a brochures. Maybe you will be reaching out to local communities where TB patients are more. You will be doing a survey, which locality you will be getting more patient. So there you will set a site, right? Because when I was having a site in Delhi, so they done a lot of research. Now Delhi is also so big that where our site should be so that we can get a maximum number of infants. So how they came to know? By birth rate. So that is your strategy. But you need not to work. You will ask your investigator, they will give you the data. Allocation of a study subject to group, like, will it be randomization 1 by 1? Study drug preparation, like how study drug will be prepared. Is it a drug? Is it a capsule? Is it a IV? How you will be administrating? Standard of care, care. Any comparative treatment is being given. Contraindication, deviation, any stopping rules. Suppose most of the protocols it is written, if SAE happen, drug will be stopped. Is that line is written in your protocol? Schedule of visit, I will show you this in a practical way. How blinding and unblinding will be measured? Study endpoint assessment. Say we have, for a safety we have said AEs will be measured. So how this they will measure this. How they will assess? They will tell a patient that if you feel if you felt uncomfortable, if you feel sick, just inform. And the moment the patient inform them, they will report as an adverse event. Study treatment. What treatment will be administered? Sufficient detail on replication, including how and when they will be administered. Right? Strategies to improve the compliance. Because mostly what happens, patient they start in the trial, but midway they drop, they lose interest. That happens. Like you may enroll for some course, but during midway you feel like not for me, you drop. Like that happens in the clinical trials. How to manage a blind so there is no leak from internal team. Even details like that, how they have to visit, when they have to visit a clinic, that is always mentioned in a your protocol. Rationale and formulation, rationale for doses, you will get from your preclinical studies. Who will perform the assessment? Like doctor, which area, all that will be mentioned. Discontinuation and modification criteria and restriction. Suppose you want to discontinue the drug doses, that will be mentioned in your protocol, that if patients face some serious adverse event, drug can be discontinued. Dose escalation, re-escalation. Sometimes protocols, they start with 5 mg and they go with 10 mg, then 50. That is dose escalation. Slowly we are increasing the dose. So if it is such a protocol, that detail will be added. Any local reactions? Guideline for discontinuation of a treatment. Suppose if you want to discontinue patient from a treatment, you may be observing that patient for a 1 week, 2 week. If that kind of guidance is there, it should be part of a protocol. What are already expected adverse event like a fever, chills? From where you will get investigative brochure. Occupational safety, because these are under safety issues. Now, in any protocol, you are always assessing two things. One is safety, one is efficacy. Safety is how safe this trial is, how safe this drug is. Efficacy is it really working? Right? So you have to present safety and efficacy endpoints. Mostly these are given to you by a study team. I can give you example. The way I have given you example of immunogenicity. This is efficacy endpoint. This is efficacy endpoint in a vaccine trial, right? So they will compare the baseline with end of study value. Baseline means day 0. So they will compare your antibody count, how much it is increased. So that is your efficacy. Safety. Sorry. Your number of adverse event, serious adverse event, and even your grading of severity, intensity, or seriousness of adverse event, blinding, unblinding. Suppose breaking, the blinding has been broken, maybe because of the team. So that they have to, that you have to already give a precaution in your protocol. What are the stopping rules? How assay will be reported? Like within 24 hours, assay is to be reported. That timeline also be mentioned in the protocol. Mostly these are type of things is copy paste. Like because this is assay criteria is same, it is going to be same only, right? But little detail maybe you have to add. Like one of assay criteria is hospitalization. Suppose you are doing a trial on infants, that hospitalization is 6 hours only. 6 hours will be considered hospitalization. Generally, it is at 24 hours. So these type of variations that you have to, adjust, but otherwise most of the section is copy paste. So this is a flow of activities which is included in every protocol. As a medical writer, you include it in your protocol, but if you are a CRC, you take a if you are not able to read your whole protocol, take a print out of this and paste in your, in your working desk or keep on your laptop. This will tell you when and what will happen in the clinical trial. This is a snapshot of the whole, plan. See, consent will happen. So this is what, this is dummy only, okay? On day one, so D1. So it has a minus 40 thing, means they have a 15-16 days, they can complete it. Here, consent will happen. So after that, consent will never, is not happening till follow-up. See, it's a blank. So inclusion and exclusion criteria, they will be checked all the time till the randomization. Medical history, screening time, they are checking. Physical exams, see, every time, screening, run-in period, randomization, treatment. So when that, what is this meaning? That whenever patient is coming to clinic, they are checking the vitals. So now I want adverse event, because this is end point of a safety. No concern and screening run-in period. From a randomization, they started checking till follow-up. Follow-up means no drug is given, but they're following up patient for a safety for 6 months or so. See, this is subject global assessment quality of life. That means efficacy. They will be measuring, is it working? So they are measuring screening time and treatment, because this is related to efficacy. So my whole point to tell you, if you wanted to start understanding protocol, download any protocol and go to study flowchart, also known as a study schema. So you'd go there and try to read like when and what they are going to do it. Even suppose you have joined as a CRC, CRA, data associate, PV associate. If you don't have a time to read a whole protocol, just go there and try to understand what protocol is saying. Now, ethical statistical data management consideration. Ethical, mostly copy paste. Mostly we are having the same because we are following ICH. It is by IRB and Declaration of Helsinki. We will also be listing informed consent procedure, waiver of consent, additional consent. These may vary little bit as per, study. And for one study, it may be different, other study may be different. But a lot of information remains same. Statistical, what hypothesis you are testing, what is sample size, what is assumption, what is power calculation, endpoint, interim analysis, how patient can withdraw and discontinue, analysis population. This is interesting, analysis population. And on which population you will be doing a trial. Suppose there are 10 patients, those passed the screening, but after randomization, they didn't take drug. So that means no point to analyze these subjects. So that is always defined by STAT. The patient who have taken at least one dose. So who will give you lot inputs from this section? Study statistician. Every team has a study statistician and a programmer on the board. Data management. Your data management team will be helping you here. How data will be collected. How quality will be maintained. Monitoring, because that monitoring is important, that how you are going to ensure that the right safety well-being of the subject is protected and the data is verified, right? When the data lock is going to happen, any retention and follow-up, deviation, audit and inspection. Again, a GCP principle, right? So now this, we are moving almost into the last section of a protocol. What is protocol synopsis? Anyone? What is abstract? Because maybe you have not gone through some protocol, but you must have read some manuscript. What is abstract? Anyone? Write your answer in the chat. No one? That's bad. Anything, right, wrong, doesn't matter. Summary, good, try, yes. Anyone else? Okay. Short summary of study which attract readers. Brief description of clinical trial. Okay, good try. Partially your answers are correct, right? Now we have studied so many sections like study objective, endpoint, study population, eligibility criteria, methods, analysis. We have gone through a lot. Our protocol may be of 150 pages. How about you don't have that much of time? One want a summary, quick, as one of you said, summary. So the only difference is it will be having a summary brief of each section, each important section. Like we can ethics section, monitoring, you can drop. So it will give you brief on idea and introduction. Study objective is very, very important. Endpoint, study population, methods and analysis. So when you write synopsis, do you write before writing full protocol or after writing full protocol? So when you will write study protocol synopsis, after writing full protocol or before writing full protocol? Your answer, please. After, very nice. After writing, yeah. Because now you have a full information. That's why I have also included after. Although it is added after the table of content, it is added there in the protocol. I will show you the actual protocol. But it is drafted after once you have written whole protocol, because then it's easy. You already having the whole information with you. Now you just have to write a synopsis and add it. Now there will be few appendices added in the protocol. What it is? Either investigator brochure, or you can say device brochure if it is a device trial. Drug information sheet, IP label, diagnostic and treatment guidelines, questionnaires for study related procedure, like example, quality of assessment, patient diaries, advertisements, lab and operation manuals, lab processings, informed consent form. So informed consent form is developed along with the protocol. If you are working on some grant, that CRF, manual of study, they are all added in the protocol appendices. This I can talk about little bit, but not much. So what happened if you wanted to see how does medical writer fits? Generally, any medical writing document, it have at least 2 cycles. So maybe study team or a sponsor, they will give you the source documents. You will be doing a kickoff meeting. to understand the expectation. Then, based on all the source document and kickoff meeting, you will make a draft one. That is here, first draft stage. Based on your therapeutic area, drug development, all the document shared with you, you will draft one. Then internal QC. Then you will send it to review round 1 for a study team. They will give you comment. Based on these comments, you will make a draft 2. Draft 2. So based on the inputs from sponsor and based on all the people who reviewed, populate all other general and specific sections in the protocol, basically address their concern. Now, draft 2 also, after this, there will be a review round 2. So 2 review round 1 here and 1 here. Then there may be further question. You will address it again, then it will become a final. See, when you're making a final draft, ensure the corrections in the information and everything is basically correct. Final draft will go for the approval, internal approval, sign off. And this cycle mostly happens for almost every medical writing. In between, there may be a meeting. So how it happens? So draft one, internal QC, review round 1 by study team. Then draft 2, you will be working, you will be addressing all comments. Then review round 2, again study team. Then final. Then approval. Okay, so this is the minimum cycle goes. So this internal QC may also again happen here also. Just to help you with the quality of the document. Why I'm telling you internal QC again and again, most of the medical writers, when they get their first medical writing job, they often give up the role of a QC instead of a writer. That's also very interesting because you will learn a lot during QC. Okay, so that's all for a protocol. Now I will give you some practical example. I will tell you how you can download from a registry, right? And can use it for your own as a health self guide. Just give me a moment. I will reshare the screen. Okay, let me share my browser. So, one thing I will recommend you all guys, make a notes. Bring your notebook, copy pencil and follow me. So this is your, I am here in a browser. So here what you do is just type clinical trials.gov. I can give you link that as well. No problem. This website, not for you as a medical writer, but also if you are working on other roles, you even working as a freelancer. So freelancer, if you're working, and this site will be working as a mentor for you. So now here you can download protocols as well. First of all, let's see And moreover, if you are finding that I can't understand protocol, so here the summaries are posted, protocol summary we post, right? So try to understand that summary, then slowly you can move your next step to protocol. So now here, if I am writing RSV, because now lot many trials are going on RSV, right? So let's do. I'm just randomly typing. You can type anything. So now you see here it has given me 709 studies. So now let me show you how you can read protocol from here. So they always give a protocol summary. So here you will be seeing study overview. They will be giving you the overview, detailed description, small little high level detail, even key milestones, when study is starting, when you are going to complete a study. What is the MC? Look at 13,000 subjects, right? No, I think 1,30,000.
Excuse
Sorry.
me, ma'am.
Yes, yes.
Ma'am, your screen is not visible.
Not visible. Then you have to tell me. Yes. So let me share again. Huh? So we can. Oh, wrong web page. So let me go back so I can explain you again. So this is Google page. Now, can you see the browser?
Yes, ma'am.
Thank you, thank you. So here you just have to type anything which you like. So I have just typed RSP. Lot many respiratory related trial is going. So I'm just sharing with you how you can read a summary of a. So this is one trial, vaccine effectiveness. Basically, they are trying to do a study which is on a vaccine effectiveness on RSV hospitalization in the adults. So see, on a study overview, you can also turn on the researcher view also. So here the information will be a lot cleaner. So when this study submitted 24, when first posted here, all dates are here. What is the primary goal of this study? That is mentioned here. Hospitalization for RSV related respiratory tract disease. So that is their primary goal, right? Secondary, in a secondary that they have mentioned here. Basically, what are the goals and endpoint that they have mentioned? Now, what else you can see here? Let's go back to this side. I can switch. So from here, you can even switch like a patient criteria. What is eligibility criteria? Very easily you can read here. See, eligibility criteria 60 years and older, adult. See, inclusion exclusion criteria, they have given such in a simple way. Age 60 years above, they should be signing informed consent because this is phase 4 type of study. Exclusion criteria. For this, there is no specific such criteria. How this study will be given? See, these are arms and group. One is they are giving RSV vaccine. Another is they are not giving any vaccine that will act as a control. Basically, they are having a two groups. Arms means group. They are having a two groups. One they are giving RSV vaccine. Another they are not giving anything. So these are your goals, study endpoints. Hospitalization rate for 14 days. They are checking. This is your primary goal. This is your secondary goal, right? So this is how simply they add. So this is what they can do. So now more information. So here more information. You can also download the protocol also for this study. If they have submitted the result, then it will be there. Otherwise, no. So they have not submitted the result. So as of now, their protocol is not available here. But I can show you for other studies how you can do it. So what you will do, you will come here again. Now from your left side, keep scrolling. You can, from here you can do anything like select interventional, or if you wanted to understand just observational, select observational, you can apply anything, right? So but main thing is I wanted to show you that we wanted to download protocol. In fact, you can also download the informed consent form. So let's stick with the protocol because that's our today's session. Apply, so it will, you will find in the study document, apply filter. So all the studies with the result, they will be having a protocol also. Let's first study, click. From here also, you go to more information. See, here is your protocol. Here is mentioned. Even amendment letter, stat analysis, all is available here. So let's download this. So tomorrow, if someone is giving you protocol writing assignment, your first job is come on clinicaltrial.gov and try to download the similar protocols. You will be it will be helpful. See here, this is the phase, this is the title of your protocol. See, randomized phase 1/2 study for safety and immunogenicity, some vaccine trial, right? Who is sponsor? Some national research institute is the sponsor, right? So even all the authors and everything is written here. See, table of content. We have learned about introduction, overview, background, everything is there. Objectives, study design and population, study consideration, study product, study visits, how one will be, see the detailed acute phase, contact, daily visits, everything is there. Safety assessment, how they will be assessing the safety. See, expedited adverse event reporting, every detail you will see. Participant management, management of adverse events, even statistical consider, why this is not highlighting, stat consideration, how and everything will, primary, secondary, exploratory endpoints will be measured, monitoring, when monitoring will be happening, data handling, number 10, clinical site monitoring, ethics, human subject protection, which is ethics committee, administrative procedure, regulatory oversight, protocol registration, a publication. What is your plan for a publication? Mostly standard for each pharma company, it is a standard. And finally, references. So I will strongly recommend that you download 1 and 2 protocols and then you read. And if you get some assignment, read here. Even you are, suppose you got a job for a CRC, you know this hospital is working on a oncology trial. Download 1 or 2 oncology protocols and read little. And if you, even if you don't want to download, it's okay, read a protocol summary. See, I showed you that workflow, they always are having this schema of, I think I won't be able to download it, so go directly because I'm working on this device. So after abbreviations, you will be seeing the, I believe here, protocol synopsis. This is more an administrative. See, this is how they have given a study overview in the form of a diagram. This is schema they have included. Like we are enrolling 130 patient, 40 will be going here, 40 will be going here, 10 will be going here, 10 will be going into placebo. So everything they try to explain with the help of a diagram so that you can understand easily. See, day 0, study product administration, day 28, blood collection, day 56, blood collection in a RSPCG. Even for a weekly contact, they have like November, December, Jan. They try to explain everything. So it is very interesting. So I'll recommend download 1 or 2 protocols and go through it. Okay, do you want me to share a link? I can share a link with you for clinicaltrial.com. Okay. So now we can also discuss the question answers. Okay. So actually what happens, this is good thing that someone they unmuted themselves and then told me that screen is not visible. What happened when I am sharing my screen? I am not able to read the messages. That is the irony. That is the issue. So the moment either I have to switch a screen and have to come here and read the messages, or otherwise the way you helped me today, that unmute yourself and tell me that screen is visible or voice is not audible properly, whatever, always unmute yourself, then tell, give the feedback. Okay, any question? Is protocol is Are you able to understand the protocol now, or it seems like a difficult? Okay, okay. Okay, then please go to clinicaltrial.com. I will also recommend go to CTRI. Hamar, it is Indian website. Also saying that go to Utericity. It's a European register. So it's important. It will give you the feel, right? That how different registries they work, right? Okay, then if there is no question, then we can close. Yeah. Take care. Bye bye. Thank you.